Cell cycle can happen anywhere in your body. It begins in the G1 phase, where the cell grows. Next, it enters the GO phase, where the cell lives. Then, it goes into the R stage, which is the point of no return, and the cell must go through mitosis. After this is the S phase, where DNA is replicated. Then comes G2, where the cells prepare for division. All of these stages lead to mitosis, which is also known as the M phase. After mitosis occurs, the cell cycle is able to repeat itself. And only eukaryotic cells go through this mitosis because it is the division of the nucleus.
Think about it this way. Start with a town. The town begins to grow as more people enter it. As it grows to a sufficient population, the town prospers and it stabilizes. The people in this town then are under a new government system, so people begin to notice that they have different views, but they can't live together. The town has to split into two different groups, forming two new towns. After this happens, this cycle will most likely repeat itself in the two new towns.
The most important phase of the process is the M phase. Mitosis takes place everywhere, but the reproductive organs, like sperm cells. The first part is interphase, but it is not one of the 4 steps of mitosis. Prophase is step 1. DNA coils up to form chromosomes. Chromosomes are the structure of DNA and they contain genes. Homologous chromosomes are chromosomes that have the same genes on them, and in mitosis they use diploid cells because of these homologous chromosomes, since the genes come from both a mom and a dad. The diploid cell has 2 sets of 23 chromosomes that sum to a total of 46 chromosomes. In this phase, the nucleolus and the nuclear membrane also break down. Centrosomes appear in animals that have centrioles. After this happens, they travel to opposite poles with spindle fibers.
Next phase is metaphase, where the kinetochore fibers move the chromosomes to the center of the cell. When this happens, it's easy to see that the chromosomes form a line in the center of the cell.
Next comes anaphase. Chromatids separate and move to opposite poles of the cell. Chromatids are the replication of chromosomes. A problem that could occur is if a chromatid didn't efficiently separate and one cell gets 2 of the same chromatid.
The last phase of mitotic cell division is telophase. This phase takes the shortest amount of time. Here, chromosomes uncoil and spindle fibers break apart. A nuclear envelope forms along the formation of a nucleolus. The result of this whole process is that there are 2 daughter cells that form, which are identical. The goal is that there's the same number of chromosomes in each cell that the cell originally started with. Meiosis is the nuclear division that has the initial goal of reducing the amount of chromosomes in a new cell. Haploid cells are used here because they deal with sperm cells. The green strands that are shown are the 46 homologous chromosomes.
In meiosis 1, it begins with prophase 1, and the only difference between this and mitosis prophase is that here a synapsis occurs, which is the pairing of homologous chromosomes.
In metaphase 1, the homologous pairs randomly line up at the center of the cell in 2 lines. The spindle fibers from a pole grasp onto one homologous chromosome, and independent assortment can occur where mom and dad's chromosomes mix.
In anaphase 1, the homologous pairs separate. Go.
Telophase 1 is the same as telophase in mitosis, where the chromosomes uncoil. The result of meiosis 1 is that there are now 2 haploid cells and aren't they both aren't identical daughter cells. Now, there are 23 chromosomes, which is half of what the cell started with. In meiosis II, there is no interphase or DNA duplication from meiosis I. This stage of cell division happens in each of the two cells previously formed. All four phases are relatively the same as mitosis, and in anaphase II, spindle fibers separate and pull the chromatids to opposite poles as the other phase. The results after telophase II is that there are now 4 haploid cells with 1 strand of DNA. Now there are 4 different cells. Not every cell division process is efficient because some problems can occur. Crossing over can happen, which is when not identical chromatids combine and parts of them break apart and exchange. Here, genetic variation can take place.